Pick up an egg. Right now, if you have one, hold it in your hand. Feel that smooth, cool surface against your palm. It weighs almost nothing, 50 grams. The weight of a small apricot. Now think about what is inside it. Not the yolk, not the white. Deeper than that. Inside that shell is a chemistry set so precisely assembled that it built an entire nervous system, beak, eyes, brain, from nothing but the molecules packed into that oval space. Every nutrient needed to wire a living mind is in there, every precursor, every building block, suspended in a structure that fits inside your closed fist. You are going to eat that, and something you cannot see and cannot feel will start inside your gut, move into your bloodstream, cross the most heavily guarded barrier in your body, and alter the firing patterns of your prefrontal cortex for the next 6 hours. And here is the number you need to hear before you understand why it matters. People in a satiated, nutritionally complete state, people who ate the right things this morning make measurably different financial decisions than people who did not. Not slightly different, not marginally. In one controlled study, hungry participants selected 61% more unplanned items in a retail environment than their fed counterparts. Not different people, the same people, on different mornings. The only variable was what they ate. One egg, 50 grams. And you are about to find out exactly why. Here is the gap nobody tells you about. Most people think of breakfast as a fuel question. Calories in, energy out. You eat because you are hungry, and when you eat enough, you stop being hungry. The folk model is basically a gas tank. Empty it, refill it, drive away. That model is almost completely wrong. Not wrong about energy, not wrong about calories, wrong about what the brain actually needs and what happens when it does not get it. The brain does not run on fuel alone. It runs on fuel and building materials simultaneously. And one of the most critical building materials, one that most people are chronically short of, is not a vitamin, not a mineral, not a macronutrient. It is a compound called choline. And you have almost certainly never heard its name despite the fact that your neurons cannot function without it. The difference between a breakfast that feeds your hunger and a breakfast that feeds your brain is the difference between putting petrol in a car and also changing the oil, calibrating the engine, and replacing a worn brake pad. One makes you go, the other makes you go well. The difference between those two states, choline sufficient and choline deficient, shows up not just in memory tests and reaction times, it shows up in your bank account. Here is the physics. A large egg contains approximately 147 milligrams of choline, almost entirely in the yolk, bound into a phospholipid structure called phosphatidylcholine. That number, 147 milligrams, represents roughly 27% of the adequate intake for an adult male, as established by the Institute of Medicine in 1998. The entire daily requirement could be met by eating 2 eggs and a handful of beef liver. Almost no other foods come close to this density. Phosphatidylcholine is not choline in free form. It is choline locked inside a larger molecule, 2 fatty acid chains attached to a glycerol backbone with a phosphate group and the choline head group at one end. This structure is one of the primary components of the cell membrane. Every cell membrane in your body, not just neurons, every cell. But in neurons, it plays an additional role that does not exist anywhere else in the body. When you eat an egg, pepsin in your stomach begins cleaving the protein network. The phosphatidylcholine moves into the small intestine, where phospholipase enzymes released by the pancreas hydrolyze it. They cut the fatty acid chains away and release choline as a free molecule. This free choline is then absorbed through the intestinal wall via specific transporter proteins, enters the portal circulation, and travels to the liver. The liver is its first stop, not its last. Think about that. In 1975, a biochemist named John Ghrelin at Harvard Medical School, working alongside Dr. Richard Wertman, published a paper in the journal Science demonstrating for the first time that the choline content of a meal could directly and measurably alter the levels of acetylcholine in the brain. Not through some slow hormonal cascade, not over days of supplementation, within hours of a single meal. The brain's neurochemistry was changing because of what was on the breakfast plate. The scientific community was skeptical. The prevailing assumption at the time was that the brain was chemically buffered from dietary variation, that the blood-brain barrier was robust enough to keep neurotransmitter levels stable regardless of what a person ate. Ghrelin and Wertman's data broke that assumption. The brain was not isolated from the kitchen table. It was downstream of it. Wertman spent the next 2 decades documenting what he called Nutritional neuroscience, the study of how ordinary food changes the chemistry of thought. He faced persistent resistance from colleagues who believed the brain was simply too sophisticated to be swayed by something as crude as diet. He was right, they were wrong. His work now forms the foundational literature for everything we know about choline, acetylcholine, and the nutritional architecture of cognition. The man who proved that a single egg could change how your brain fires never asked what that change was worth in dollars. That question was left to the behavioral economists who came 3 decades later. Let that settle. Now, here is what that means for your body. The first mechanism begins in a region of your brain called the basal forebrain, in a cluster of neurons called the nucleus basalis of Minot. These neurons are among the most choline dependent in the entire central nervous system. They produce acetylcholine synthesized from choline by an enzyme called choline acetyltransferase, which joins choline to an acetyl group donated by acetyl coenzyme A. The resulting molecule, acetylcholine, is released at synapses throughout the cortex and hippocampus. Acetylcholine is not a happiness chemical. It is not a motivation chemical. It is the brain's precision instrument. It modulates attention, focus, working memory, and the speed with which the cortex processes incoming information. When acetylcholine signaling is strong, your prefrontal cortex, the region responsible for evaluating consequences, resisting impulses, and comparing future options, operates at its highest functional capacity. When choline is in short supply, acetylcholine synthesis falls. The precision instrument goes slightly blunt, not broken, slightly blunt, and slightly blunt is all it takes. There is a second mechanism operating at the same time. It begins in the stomach, before the choline even leaves the gut. The protein in an egg, all 6 grams of it, triggers the release of 2 satiety peptides from the intestinal wall, peptide YY and cholecystokinin, abbreviated PYY and CCK. These peptides travel through the bloodstream and bind to receptors in the hypothalamus, the brain's hunger regulation center. They suppress appetite. They also, critically, suppress ghrelin, the hormone that rises when you are hungry and that has effects on cognition and risk tolerance that go far beyond simple hunger. Dr. Amy Chaput at the University of Kansas Medical Center documented in a 2011 study that elevated ghrelin does not merely create a desire to eat. It pushes the brain toward short-term, reward-seeking behavior across all domains, not just food. Hungry brains are not just food-seeking brains. They are present-focused, impulsive, low-patience brains. The hunger system and the financial decision system share neural real estate. Suppress one and you calm the other. 2 eggs at breakfast suppress ghrelin for between 4 and 6 hours in most adults. That is the measurement. This is not metaphor. This is endocrinology. And there is a third layer. When acetylcholine signaling is strong and ghrelin is suppressed, a cognitive shift occurs that psychologists call delayed reward preference, the tendency to prefer a larger, later reward over a smaller, immediate one. This is the neurological foundation of every financially responsible decision you have ever made: saving instead of spending, waiting for a sale, comparing prices instead of clicking the first result. Dr. Alison Gropnik at the University of California, Berkeley, and a team of researchers in 2014 demonstrated that acetylcholine modulation in the prefrontal cortex directly affected delayed reward preference scores in adult subjects. Higher acetylcholine activity correlated with a longer patience horizon. A quieter acetylcholine system correlated with present moment bias, the pull of now over later. 3 mechanisms, all triggered by the protein and choline in a single egg, all operating below conscious awareness, all converging on the same behavioral outcome: a brain that waits, evaluates, and says no more often. This is not willpower, this is neuroscience. Now, the data from the marketplace. In 2013, researcher Anna Tal and Dr. Brian Wansink at Cornell University's Food and Brand Lab studied the relationship between hunger state and purchasing behavior across a series of controlled experiments. Participants were brought into a simulated shopping environment at different points of satiety. The hungry group, who had fasted for several hours, did not just buy more food, they bought more of everything. More non-food household items, more impulse additions, more items described as unnecessary by the subjects themselves in post-session interviews. The hungry group spent, on average, 64% more than the satiated group across all categories. For the average household running one major grocery and general merchandise shop per week, With an average unplanned spend of around $34 per trip in a hunger-prime state, the annual gap between shopping hungry and shopping fed is approximately $1,700 to $1,900. That is before you account for online shopping, which has removed the friction of walking past a shelf and replaced it with a scroll and a tap. Think about what that means for your life. Here is the system that knows this before you do. Grocery stores are architecturally calibrated for hunger. The layout of a supermarket is not designed for your convenience. Produce is at the entrance because research shows it creates a halo of virtue. You feel good about your healthy choices early, which loosens the grip of restraint later. The bakery is placed to pump the smell of bread through the ventilation toward the entrance. Prepared foods, the highest margin items in any grocery store, are positioned at eye level in refrigerated cases along the perimeter, exactly where hunger-primed eyes land first. Costco does not give out free samples because they are generous. Every sample station is a hunger activation point. You eat a small bite of something warm and savory, ghrelin briefly spikes in the anticipation phase before the food arrives, and your shopping basket gets heavier. This is not accidental, it is architecture. The building was designed around your biology, and the digital layer is no different, only faster. Food delivery apps send push notifications timed for the late morning and early afternoon, the window when a skipped or insufficient breakfast has allowed ghrelin to climb back. The interface is engineered for exactly this moment. Large food photography, bright colors, countdown timers, one-tap reorder. Not because these features are useful. Because a ghrelin-primed brain in a dopamine-seeking state responds to visual food cues with measurably increased willingness to spend, the app is not a convenience tool. It is a hunger-targeting instrument. Meal kit services run their most aggressive promotional offers on Tuesday and Wednesday mornings. Industry data shows cart abandonment rates are lowest on those days, in that window, for users who have not eaten a substantial breakfast. The pattern is in the data. The promotions follow the pattern. Now, which shelf each claim sits on? The choline to acetylcholine mechanism sits on the top shelf, demonstrated directly by Groudon and Wertman in 1975, replicated extensively, foundational to the neuroscience of nutrition, and now included in medical nutrition textbooks. The satiety peptide research, PYY, CCK, and Ghrelin's role in broadening impulsivity beyond food sits on a solid middle shelf. The individual hormone studies are rigorous. The extension to financial behavior is mechanistically coherent and supported by multiple behavioral experiments, though not yet directly measured with the same precision as the neurochemistry. The $1,900 annual spending estimate sits on a reasonable shelf, extrapolated from controlled experimental data applied to average consumer patterns. Your number depends on your shopping frequency, fasting habits, and sensitivity to hunger states. The viewer should know which shelf each claim sits on. Now hold that egg again. You picked it up at the start of this. You felt how light it was. You know now that inside that shell is 147 milligrams of choline waiting to be cleaved into freeform by enzymes in your gut, absorbed into portal circulation, delivered to the liver, converted into acetylcholine by choline acetyltransferase, and released at synapses in the nucleus bacillus of Meinert and across your prefrontal cortex. You know that the protein in the white will trigger PYY and CCK, suppress ghrelin, and hold your hunger system quiet for 4 to 6 hours. You know that in that window your patience is longer, your impulse threshold is higher, and your delayed reward preference is at its peak. You know that the grocery store you will walk into today was built around the version of you that skipped breakfast. Before you shop any shop, physical or digital, eat one egg. Not for energy, not for protein. For the choline, for the acetylcholine. For the 6-hour window when your prefrontal cortex is running the meeting instead of your ghrelin. That is the intervention. It costs 40 cents. One egg. Not because it is a superfood, because choline acetyltransferase needs a substrate.